Hey listeners, I'm excited to have you joining us for episode 663 of People Behind the Science. I'm your host, Dr. Marie McNeely, and this week we are rebroadcasting our interview with Dr. Karen James, originally published on February 2nd of 2015. Now listeners, if you haven't met Karen yet, she does remarkable work that combines genetics research, education, and outreach. She uses DNA to identify species of animals and plants for a variety of projects, including some citizen science projects where members of the public have contributed. And the goal of her work is to increase our ability to understand and manage how changes to the environment affect the diversity of living things that is so important for our lives and our planet. Now, in our interview, Karen shares great stories about her life in science. So get ready to be inspired in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am excited to be here today with Dr. Karen James. So welcome to our show, Karen. How are you?
I'm great. I'm really happy to be here.
Excellent. Well, we are delighted to speak with you today. Now, listeners, Karen is a staff scientist at the Mount Desert Island Biological Laboratory in Maine. She received her PhD in genetics from the University of Washington and conducted postdoctoral research at the Natural History Museum in London before joining the MDI Lab. Karen is also co-founder and director of the UK-based charity, the HMS Beagle Trust, which is working to rebuild the HMS Beagle and retrace the journey of Charles Darwin with a new generation of students and scientists. So Karen, you are doing some wonderful things there. We are excited to hear about your journey through life and science. So today we want to get to know you, both as a scientist, but also as a person. So can you take a few minutes to tell us what you like to do when you're not doing science?
Yeah, thanks for that introduction. I'm definitely an outdoors person, and when I'm not doing science, you can find me on the trails of Acadia National Park, which kind of dominates the desert island that I both work and live on. I do a lot of hiking and bicycling and skiing, cross-country skiing, and I enjoy working in the garden. Though I don't really have a full-fledged garden, that would be the science getting in the way.
Right. Understandable.
That's something I aspire to do more of, but I love being outside as much as I possibly can.
Wonderful. Well, Karen, it is great to get a picture of what you do when you're not at work, enjoying the great outdoors, attempting gardening. I know I'm terrible at it myself, though I have tried. So wonderful to hear about your hobbies and how you like to spend your time. And now we're going to dive into your scientific side, what you do at work. So can you tell us today how you describe what you do to someone who might not be in science?
Right. So I'm a geneticist. You mentioned that's what I got my PhD in. And I'm applying that. I'm working to use that genetics knowledge and those research tools and strategies in support of environmental research and conservation and restoration. So more specifically, I use DNA to identify animals and plants, including potentially those collected by members of the public or citizen scientists, they're called, in the service of a whole range of research and conservation and restoration projects, initially here in the Acadia region, which includes Acadia National Park and the surrounding ocean and areas nearby. But my vision is to go beyond that to other national parks and maybe even the national long-distance trails eventually.
Oh, that is a phenomenal research endeavor. So do you find yourself when you're hiking through the forest, the parks, kind of always in search of new samples?
Well, you have to be careful about that because you can't just go into a national park and collect things. That's illegal. You need a research permit. So when I'm generally out hiking or biking, I don't have a blanket permit where I can just collect samples anywhere, anytime I find them. But yes, I'm always thinking about locations, especially places in the park maybe where I might get members of the public, visitors to the national park, interested in participating in science. For example, really popular trails or bike paths where I think I might be able to recruit people to do a little science while they're here for recreation or just sightseeing or whatever reason they want to come to a national park.
Absolutely. Well, that is a great opportunity to get the public, to get people involved and excited about science and the work that you're doing. So great to hear a little bit more about it. And I think with all the things that scientists like to do in a day, all the work that they're doing in the lab and all the things that they like to balance outside the lab, you mentioned a lot of these outdoor activities that you enjoy. It's hard to find enough time in the day sometimes. So can you tell us, Karen, do you have a particular motivational quote or a saying that you love to keep in your mind to keep yourself motivated and focused?
That's always hard. There's so many quotes that I've come across over the years that I like and that inspire me in one way or another. And this is maybe going to be corny considering my background, but I really like the last paragraph of Charles Darwin's On the Origin of Species, especially the part about the kind of never-ending evolution of beautiful and wonderful forms. So maybe I should read that.
Sure.
It's like the last half of the last paragraph, and it goes, "Thus from the war of nature, from famine and death, the most exalted object which we are capable of conceiving, namely the production of higher animals, directly follows. There is grandeur in this view of life, with its several powers, having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved."
Well, that is a wonderfully inspiring passage. And I think a lot of biologists look to Charles Darwin for inspiration, especially these people who are interested in the evolutionary bent of things. So phenomenal source of inspiration, Karen. Now, I'm curious to hear when I talk to scientists about who some of their role models were. So we mentioned Darwin here. Are there others, people who you looked up to throughout your journey and your career as role models or inspirational figures?
Sure. This has always been a tough one for me because whenever anybody asks me, especially when I was a young person, what teacher or other role model really influenced my decision to become a scientist, I'm always drawing a blank on that one. And I feel sort of bad. I'm sure there's some people that were very important in that way, but, I just don't have a single person who leaps to mind when I think about that. More recently, it's easier to identify some more recent role models. And these are people who aren't necessarily, role models in the traditional sense in that I can see myself doing what they do, but they're models in the sense that they have certain characteristics or ways of working or ways of being that I admire for one reason or another. Many of those role models I find are on Twitter, actually. And I would say that if I had to pick a role model, I would say the women scientists of Twitter.
I like it.
Because they kind of show me this broad picture of what it is to be a woman in science and all the different ways that how that can look. And, you have people like Kate Clancy, Danielle Lee, Hope Yaren, so many others. I could go on for a very long time. And there are a lot of lists that I've just been recently retweeting of women scientists on Twitter. And collectively, they are my role model because they show such a diversity of ways of being successful that I can imagine myself being successful in those different kinds of ways, or even in my own way.
Well, I think that is an excellent source of role models, the women of Twitter. I love it. So do you find yourself on Twitter frequently, or do you find that engaging in social media as a scientist is helpful?
Oh, yes. The people who are listening to this who are on Twitter and follow me there are probably laughing Because when you ask, do you find yourself on Twitter a lot? They're thinking, yeah, she's on Twitter all the time.
Even when sleeping, yes.
Yeah, pretty much. Yeah, I use Twitter as a source of both and a repository, I guess, for both my personal interests and also my professional interests. And there have been several papers out now talking about the benefits of Twitter for academics and for scientists. And I've definitely reaped a lot of those benefits myself, whether it's opportunities to collaborate or travel or just become more aware of certain topics or issues and general awareness of what's happening in my field and more broadly in science. It's just been invaluable to me. There are several projects and things that have happened over the course of my career since I started Twitter that probably wouldn't have otherwise. So I have absolutely no regrets about the time I spent on Twitter.
Wonderful to hear. Well, I myself am on Twitter, and it's interesting because I feel like I talk to some scientists who are exactly as you are, kind of always plugged in, always engaged, and really find it a helpful tool to connect with other scientists, to network, to even find new references, literature papers out there. And they're at sort of the other camp who is like, Twitter what? Yeah. They have no idea, and they don't necessarily see it as a valuable resource. So I'm excited to see more and more scientists getting on the social media stream here. So great to hear about some of these people who've really inspired you and helped you out throughout your career. And I'm curious, now we talked about no one particular person sparking your interest in science and getting you engaged in that scientific career, but can you tell us what are some of your earliest memories of being excited about science? Was this tied back to a particular point in your life?
Oh, that's a good question. My grandparents had a cotton farm in West Texas, and I have very vivid memories of being there and, squatting down and looking into a puddle in the irrigation ditch and finding tadpoles and collecting them and, raising them to be frogs and having them hop away, or just, being outside and seeing plants and animals and really getting literally muddy. I think that those kind of outdoor experiences were probably formative for me in terms of science and just being interested in nature and how it works. My mother tells me that I was kind of a know-it-all. I think maybe I still am.
It's okay. Many of us are guilty.
And that I would say things with great confidence, which I now know I should have called hypotheses, but I thought I was sure about them. And she said, for example, she wrote several of them down because she thought they were so funny. And one of them was, for example, that I said that a spider spins its web from its heart.
Oh, wow.
And
interesting hypothesis.
I was sure that's how a spider spun its web. And I also claimed that cantaloupes came from cantaloupe factories, which probably would have made my grandfather growl if he heard that, because he actually grew cantaloupes. So
oh, wow.
That's terrible. And then I had a fairly religious upbringing, and apparently I said something like, I don't understand why God made us with spaces between our fingers, because it's so annoying having to dry in between all of your fingers every time you wash your hands.
I love all of these little kids saying they're wonderful.
Yeah, so apparently, though I don't remember actually uttering these things, I was definitely thinking about, here's how things are. Why might that be? I don't understand. Here's an idea. That's definitely the way I was thinking even as a 4-year-old.
Well, that is phenomenal. I love to hear about how each scientist kind of got their start along the scientific route, and definitely being one of these asking all sorts of questions kids, or even I love this description of the know-it-all, the overly confident kid, kind of asserting your own hypotheses about how the world works. I think that is wonderful. So can you tell us a little bit now how you went from this very confident 4-year-old to where you are today, Karen? Some of these key moments that shaped your journey.
Well, after high school, I went to a state university, Colorado State University, and I wanted to be a veterinarian, actually, because I loved animals. And I was an intern at a veterinary hospital the summer before college. And through that internship, I realized that I think I loved animals too much, because it was really difficult for me to emotionally distance myself. So I ended up changing majors within the first few weeks of being in college, from pre-vet to, I think next I went to electrical engineering. I don't know why I picked
wow.
I don't know why I picked
So big change, yeah.
And then I switched then to biology. And the reason I did that is that as a freshman, I really enjoyed just my Bio 101 course. And also I had an honors introduction to biology, and there was a lot of kind of ethics and more philosophy in that course. And that's really what got me turned on to things like The Origin of Species and seminal books in the history of science and the philosophy of science. And then I got stuck into biology through that, but then I took a cell and molecular biology course, I think as a sophomore, and I was so kind of awestruck by the intricate workings inside the cell, the way that the DNA is transcribed into RNA and then translated into protein, and then those proteins are doing all their things that they're doing. And it just was this amazing, whole different world that I'd never really seen or known about before. And I loved that so much that I took the opportunity that my professor who taught that course, Tom Wilson, offered to have some undergraduates work in his lab. And so I worked in his lab for over the course of about a year and a half for the rest of my stint in college. And I started doing genetics with Drosophila fruit flies. And I really loved genetics. And then he encouraged me to apply to graduate schools. And I loved the idea of continuing with research. So I applied, and I went to the University of Washington, where I continued in Drosophila genetics. And I worked on developmental biology and genetics. I was working on patterning of the early Drosophila embryo and the eggshell that surrounds it. And then towards the end of my PhD, I got really interested in evolution. In particular, I was trying to move my project in an evolutionary direction by exploring the possible underpinnings of the differences in the patterning of these embryos and eggshells between different fruit fly species. Because some eggshells, Drosophila melanogaster eggshells, have these two dorsal appendages, two little prongs that stick off the eggshell. Mm-hmm. But a related species, Drosophila virilis, has 4, and another species has 6, and another one has 8, and another one has 1, and there are a few species that have 0, and it just seemed like such an interesting potential model for understanding evolution. And so at the end of my PhD, I wanted to continue that move towards evolution from development. And so I was looking for postdoctoral positions in evolutionary developmental biology, or EVO-DEVO. And at the same time, I moved to London from Seattle with my partner. He is also a geneticist, and he had a postdoc opportunity in London, and we moved together, and I was looking for postdocs too. After a fairly long job search, I found this postdoctoral position at the Natural History Museum, which, considering my kind of interest in evolution and the history of science and philosophy of science that I'd become interested in college, was just kind of a dream come true to be able to work in and walk the halls of the museum and meet all of the amazing people there. And I started working more on evolutionary developmental biology in plants. I was in the botany department. And then in 2005, as a member of the department who had an interest in genetics, I attended the first international meeting for the Barcode of Life, which was the first meeting of a group of researchers interested in developing a DNA-based system for identifying biological specimens to the species level. And this seemed like an obvious thing for me to be involved in, since I had a genetics background, and I was looking at questions of biodiversity and evolution and natural history. And so I really got stuck into that area, and I've been working in that ever since then. And at about the same time, I became the coordinator for the upcoming Darwin's 200th birthday celebrations, which the museum was kind of the epicenter of that in the UK. And I organized a whole series of projects, and I helped in some cases actually carry out those projects, or in some other cases just facilitate them, that had some relation to Charles Darwin's legacy, either extending that legacy or working with the specimens that he had worked with or things like that. And so the last few years at the museum were really a combination of Charles Darwin's legacy, research projects related to that legacy, and then research on this DNA barcoding, this DNA-based species identification project that I mentioned. And towards the end of my time there, I was trying to get funding for my DNA barcoding project to assemble a reference library of DNA sequences from British trees. And I was having trouble finding funding for that through the kind of traditional sources. And I met up with the director of an educational charity, Adrian Richardson, the director of the Cott Hill Educational Trust. And he and I kind of made a deal that he would help fund my research project if I would engage his school children, his students, in the project. And that's kind of how I got involved in citizen science initially. So I started working with 11 to 14-year-old school children, getting them working in a lab that I set up in a renovated school building in Dorset, and got them outside collecting specimens and extracting DNA and doing, lab work to generate these reference DNA libraries. So now that I'm over here at the MDI Biological Laboratory as a staff scientist and member of faculty here, I'm continuing that sort of work, combining DNA barcoding and other DNA-based identification technologies or applications with citizen science to try and address some of the big environmental and ecological questions that might be addressed by those two things in combination.
Karen, it sounds like you have had a very exciting journey through science to get to where you are today. And I love hearing about these shifts in scientist focus. So mentioning that you were interested initially in veterinary medicine, you had that brief stint in electrical engineering, back to biology, and then really honing in on your current interest, this genetics, the DNA, the fascination with how all of that process works. So wonderful to hear about your journey and some of these projects that you've worked on over the years. And I'm curious to hear a little bit more detail now about one of these projects that you're working on that you are the most excited about, and it can be related to this DNA barcoding.
One of the things I'm really excited about right now is that I'm in this kind of an alpha testing program for a new, very, very small DNA sequencer made by Oxford Nanopore, based in the UK.
How small are we talking?
It's called the MinION, and I've got it at my desk here. And it's about the size of a cell phone.
Really? That is phenomenal.
And I got into this kind of testing program. The MinION sequencer is not yet available for sale, but they have a group of, a couple hundred scientists who are working with it to pilot it and find out what works and what doesn't and make improvements and feedback to the company so that they can improve the device and get it ready for more widespread distribution. It really is tiny compared to the big desktop sequencers that we've been using for the last several decades, really. What I've been doing with it is, as a kind of a pilot project, is using it to try and explore some of the ways that we might use it in DNA-based species identification, DNA barcoding, or other kinds of DNA-based species identification. And so this summer, I got my two high school interns, Hannah Newhall and Anna Ramgren, involved in this project. And we were collaborating with one of my colleagues here at the MDI Biolab, Dr. Jane Disney, who works on restoring eelgrass habitat in Frenchman Bay, which I can literally see from my office window here. It's the bay just north of Mount Desert Island, and it has a lot of eelgrass habitat in it that's really ecologically important, but it's been dying back over the last decade or so, and they're trying to explore and understand why that might be happening. And so I got my interns out there in the bay collecting invasive green crabs and figuring out how to dissect out their stomachs and get their stomach contents into a tube, and then using the stomach contents to make DNA, and then using the DNA in this little Min ion sequencer to see what the invasive green crabs might be eating.
Interesting.
And we're still working on, the data analysis and improving our methods because, as I said, this is kind of an alpha testing program. And so there's a number of kinks to be worked out. But it's a really exciting project, both for me and I also think for my interns who got to do both some traditional field work, getting out there, collecting green crabs, and doing some dissections in the lab, and then doing some pretty high-tech DNA sequencing too.
Well, that sounds like an excellent project and a really unique opportunity for these interns to be involved in. So very exciting. And I think there is a lot of hype about this DNA barcode concept, this project that people are working on. Do you have a sense of how many species now have been collected as part of this DNA barcode library?
Sure. There's a website, barcodinglife.org, where all this information is accessible. And I don't know the exact number right now, but I think it's getting close to 3.5 million reference DNA sequences from known species. That's things that have been discovered and named and described already by scientists over the last 300 years. And it would seem that 3.5 million is at least double the number of known species, which is around 1.5 million. But you have to remember that in this reference library, you want multiple specimens per species. So the 3.5 million sequences correspond to fewer than the total number of species that we know are out there. So there's still work to be done.
Certainly. Well, this is a fascinating area of study and really interesting to hear about your part within it. So I think so far, though, Karen, we've talked about all the great aspects of careers in science and your journey, some of these amazing projects that you're able to work on. We haven't yet talked about some of the challenges. And I know every scientist faces challenges, there are obstacles along their path. And I think it maybe even comes in waves. You have these days, weeks, maybe even longer, where you just feel like everything's going wrong or you are really struggling with something. So can you take us back in your career to a low point, to a point where you were struggling or maybe had a major failure, and tell us what happened and how you got through it and were successful?
Well, first of all, I would just say that being successful in science has a lot to do with talent and hard work, but perhaps more important is luck and privilege as well. And so I think some of the challenges that people face generally are going to be strongly influenced by luck. And for me, that was certainly true. So when I was doing my PhD, I was trying to analyze or assess the expression of this gene called PIPE in response to removing another gene called RAS. And to do this, I needed to do this protocol called an in situ hybridization. And you don't have to worry too much about what that means, just that it takes generally about a week to do it. And
it has lots of steps.
Yeah. And I could not get this thing to work. I tried so many different permutations of the protocol, so many different starting reagents. I think I got my PhD in troubleshooting, actually, not genetics.
Right. Sometimes they're synonymous, right?
This went on for, I want to say, 1.5 years, this troubleshooting of this one protocol.
Really?
And it was really hard. At some point, I definitely wanted to just give up. After about 6 months, I thought, can't we move on to something else?
Right.
Or should I change projects? And in the end, I never did get it to work. I came up with a different way of asking the same question, which involved a slightly different protocol and a slightly different approach, but asked the same question: how is the expression of this gene PIPE affected by the absence of this other gene RAS? And in the end, getting through that was about changing approaches rather than just persevering. I think I would still be working on it today.
Right.
If I hadn't switched over.
Perhaps the longest PhD in history right there.
Yeah.
Well, Karen, it's wonderful to hear about one of these tough times because I think a lot of young scientists go through these difficult points and they're struggling. Maybe they're in month 6 of trying to get something to work, doing that troubleshooting, and they feel like they're alone. Like, could anyone possibly have had the same experience? And I think so many times the answer is yes. And these people have gone on to have successful careers in science. So wonderful to hear about how you solve this problem, finding a different approach that you could use to ask that same question that did not involve this procedure that was really, really troublesome at the time. So great to hear about how you turned that into a success for yourself. And I think scientists keep so busy in the lab doing research and trying to balance all of the other aspects of their life, things that they like to do on the side. And I know I've chatted with a lot of scientists who are quite avid readers, and I love to get little recommendations of books that scientists have read and really enjoyed. So can you share one of these with us today, Karen, a book that you've read and loved that you would recommend for our listeners?
First of all, I should say that I don't read as many books as I used to. And part of that might have to do with Twitter, but there is one book that I read when I was in graduate school that really, I don't know, it felt like a scales falling from the eyes kind of moment. And it was The Botany of Desire by Michael Pollan. It's a book that's divided into 4 sections, and each section is about a different plant. And it's kind of a natural and cultural history of each of those plants. And the premise of the book is the idea that we like to think of ourselves as having domesticated plants and animals to suit our purposes. But it's equally useful and perhaps equally valid to think of the plants and animals as having domesticated us and made us perform certain tasks for them and help them survive and succeed in, the survival stakes of evolution. And it just turned everything upside down in thinking about humans' role in nature. And for me, I should say, I don't think it necessarily did that for everyone, but for me, it was a big deal to start thinking about this. And it really made me internalize the fact that we are part of our environment and that we are one of millions of species and we are interacting with the other ones. And it's really happening and we don't know the outcome. And it's a really big experiment, basically. So that book definitely was important to me. Also, it was important to me because Michael Pollan is a fantastic writer, and that was around the time I became interested in science writing. And so it was inspirational for me to read such good writing.
Absolutely. Well, it sounds like a great recommendation. And I love these books that really shake up how you think about the world and make you start questioning things just by presenting this interesting new perspective. So I will have to check this book out myself, and I will put it out on our resources tab for our listeners to find as well. And I think throughout our discussion, we've mentioned sort of inadvertently a few of these travel opportunities that you've had in talking about your training for your PhD, your postdoc, and where you are now. You've been to a few different places. And I think scientists through training, through conferences, through collaborations, get to travel actually quite a bit as part of their careers. And I'm always curious to hear about the favorite places that scientists have been to. So can you share your favorite place that you've traveled to for science with us today, Karen?
So this is pretty easy. The answer is Galapagos.
All right. I definitely saw that coming.
You probably could see that coming.
So tell us about your experience there.
Yeah. So when I was at the Natural History Museum in London, one of the projects I worked on associated with Charles Darwin's 200th birthday was a project to extract DNA from 2 of the bird specimens that he collected while he was in Galapagos. And these are specimens of a species of bird that is extremely rare. It's one of the rarest birds in the world, the Floriana mockingbird. And the bird is now extinct on the island where Darwin originally collected it. And it's surviving on 2 neighboring satellite islands. And I was involved in this population genetics project to add these 2 Darwin specimens to the larger collection of more modern specimens to help understand how to restore or how to reintroduce this bird species back to the island where it originally lived and where Darwin first encountered it. So because of that project, I became interested in Galapagos, and also through the whole Darwin 200 experience and also my role with the HMS Beagle project, I became aware of an opportunity to travel to Galapagos with a group of high school students that had won a competition run by the Wellcome Trust. This was going to be completely paid for by the trust.
Oh, wow.
And I would be serving as a kind of scientist and creative guide for the students while we were on this trip to help them interpret and capture and make meaning out of some of the experiences that they were going to have. So I applied for this opportunity and I got it, and I was just over the moon about it. And I got to go there for 12 or 13 days. And we visited 4 different islands. And one of the highlights for me was seeing the very first landing site, the original landing site in Galapagos on San Cristobal Island, the ship, the Beagle, that was carrying Charles Darwin, and the place where he was first rowed ashore in Galapagos and had his first impressions of the landscape and the species there. And to be able to stand on that same beach and think about that was definitely a goosebump-raising moment for me. And also just being able to see some of the spectacular plants and animals that live there and nowhere else on Earth is completely a dream come true. And for me and most people is not something that is within the realm of financial possibility normally. So this was definitely a trip that I only got to do because of my career and where it had led. And I probably wouldn't have been able to do otherwise.
Well, that just sounds like an awe-inspiring opportunity. I think being able to see the Galapagos almost through Darwin's eyes as you're looking at this initial spot where the Beagle landed and stepping off onto the beach, looking around. I think the Galapagos are such beautiful islands. I've been there myself, just being able to see the animals up close and personal way more than you expected you would because they're so docile, I think is just an indescribable experience.
Yeah, I agree.
Well, great to hear about this place that you were able to travel to for science and sort of the things along the way that led up to that amazing opportunity. And I love talking travel with scientists. And I think when you talk about these travel opportunities and you even think back on all these different travel memories that you have, a lot of time the people are what springs to your mind in addition to the place. And I think scientists get opportunities to work with some phenomenal people along their journey in these different lab groups. And a lot of times they're very creative and funny and just amazing people to work with. And I think as scientists, we're very serious about research, but we're also quite a bit of fun. We're people too. We like to do things that entertain ourselves. And oftentimes some different quirky traditions spring up in labs, or you get some just hilarious memories you can look back and laugh about. So can you share with us today, Karen, one of these funny science memories or even a quirky tradition that you've experienced in one of the places that you've worked?
This is kind of a tough one for me. I agree with you that there are some amazing, creative, funny people, and I've definitely encountered many of them, but it's hard for me to think of a tradition, but I can think of one story. When I was doing my graduate work, University of Washington, one of my lab mates and a dear friend of mine, Jenny Dorman, we sat back to back in this room without windows for, I was there for just over 6 years, and I think I was in that room pretty much the whole time. So no windows for 6 years.
Sounds like torture for someone who loves the outdoors as much as you do.
Well, fortunately, I was not too far from the door, and the door led to a really nice running and biking trail that was nearby on campus. So it wasn't as bad as it sounds.
Gotcha.
But we had these long, dark, rainy winters in Seattle, of course, and this dark room with no windows in these dark, rainy winters was pretty much unbearable. So we decided that we wanted to paint the room a really bright, vibrant orange color, but we knew we wouldn't be allowed. So we waited until after dark one night, and we snuck into the lab with our buckets of paint and our paintbrushes, and we completely painted the back wall of the lab this bright sort of solar orange color.
Oh, wow.
And the next day, everyone, came into work and walked by the door and jumped back and said, wow, what happened in there? Oh, we don't know.
It was like when we got here, right?
Some little gnomes must have come in, So we were definitely modifying our own environment.
So did it work? Was it cheerier in there afterwards?
Yeah, it definitely was.
Good. Well, that sounds like a great solution to the problem. I think a lot of scientists end up in these labs. They're sort of dark, almost cave-like, and you're there for so much of your time during your PhD work that it's nice to be able to bring in a little splash of color, a little bit of brightness. So great memory there. Now, did you guys end up getting in trouble for it?
Nope, we didn't.
You escaped.
We got away with it. So everyone listening to this, Karen James says you will get away with it.
Right. I love that. Absolutely. Well, that is a great story and certainly a bonding experience, I'm sure, sneaking in with your paint buckets and trying to quickly paint the wall before anyone noticed you were there.
Yep.
Well, I love to hear about these funny stories and traditions that scientists have over the years. And I think in addition to these amazing memories that you form with your collaborators, you have a lot of time to sit around and discuss some of these big questions in science. And we've talked about some of the work that you're doing now and things you've done over the years, but we haven't yet chatted about these big questions that you are dreaming to answer, Karen. So can you tell us today if things like funding and staff and technology and feasibility, all these restrictions on research, were not an issue? What is one of these big questions that you are just dying to know the answer to?
Well, I think the biggest question that is the guiding goal of everything that I do now is the question, how will environmental change, so climate change and other kinds of change, especially anthropogenic change, impact species over the next, couple of centuries or so? Can we predict that? Can we then use those predictions to change our activities, predict future problems before they happen, and avoid some of the elements of the potential catastrophic effects of climate change and other change? And so I really would love, for example, to be able to generate, if I had all the resources in the world and all the time in the world and human resources in the world, I suppose I would like to create a interactive global map of all species in past, present, and future, and be able to press play and see how they all move and respond and change when you tweak the aspects of the model that you might be working on. So, what if climate change happens this way and ocean acidification happens that way and the human population becomes this big? Where will all these 1.8 million species be in 20 years? And, where will the invasive species be? And I think being able to generate a resource like that would be just profound and incredibly needed.
Certainly. Well, I think you have drawn a marvelous image for us of what this product could look like. Just having this interactive map. I'm envisioning a lot of lights, a lot of color. I like it.
Yeah.
Well, Karen, I think that is a big question on a lot of scientists' minds right now. Thinking about environmental change in particular, how it's going to affect these species, I think is an interesting question. And I like this idea of being able to visualize and predict what is going to happen to different species with different changes that we might see over time. So fascinating question to ponder. All resources aside, I think we could get there someday, right?
Well, I think part of my vision there is that to get there, you would need this kind of unlimited human resources, because collecting all the data for generating that model, that interface, is something that even if every single scientist was a biodiversity scientist or a taxonomist or an ecologist, we still probably wouldn't have enough people. So we need to engage non-scientists in collecting data, and that's definitely an area that I'm working on that I think has a lot of potential in trying to take the next step in being able to gather and analyze data on a scale that we haven't been able to do before.
Absolutely. I think there are some great opportunities for, as you described them, citizen scientists to get involved, to be a part of this, and to contribute in a big way, just with the manpower that is needed to put this thing together. So wonderful to hear about one of these big questions that ties in nicely with some of the work that you're doing. And I think when you think back on your career, Karen, as a scientist, you think back about the people that you've worked with, the mentors that you've had. There's a lot of great sources of advice. So can you share with our listeners today one last piece of advice or maybe a piece of inspiration?
One thing that I keep coming back to myself is this commencement address by Eli Pariser, who's, I think, was the founder of MoveOn.org. This address, I think, is from 1996. So, wow, MoveOn.org, is that old? Okay.
That doesn't seem like long ago, but I guess it was.
Well, I guess it is.
Yeah.
So he gave this commencement address at Simon's Rock. He was talking about the idea that there is no one at the top who has some secret knowledge or answer or experience that they know what they're doing. We sort of imagine there are these authority figures above us, maybe, who know exactly what they're doing. And that we could never do those things because we would be clueless and not know what we were doing. So his speech was about realizing that that is not true and that we're all just kind of making it up as we go along. And he says, and so we come to the question of who is going to jump into the driver's seat and save the day. If the world doesn't run in an orderly fashion, one might conclude that it's just the wrong people are in charge. I say that with minimal partisan implications. Someone needs to make sure the trains run on time. We need to find that person and tell them to step two. But that person ain't coming. There are no experts, no heroes. It really is just us chickens. I've had the opportunity and the honor over the last 5 years to meet some of the great political leaders of our time, men and women who have spent decades fighting injustice and winning. By their accomplishments, they are extraordinary, and I do not believe it diminishes them to say they are also quite ordinary. They are, every last one of them, normal people. People who have strong suits and weak suits, good days and bad days. We fool ourselves about this sometimes. We don't want to see our heroes in the bathroom. And when we confront the great and mighty Wizard of Oz, the bellows of smoke, the flashing lights, the noise may confuse us. But when we peek behind the curtain, we find the wizard is just a man. You're a very bad man, Dorothy says, shocked. And the wizard replies, oh no, my dear, I'm a very good man. I'm just a very bad wizard. And so it goes with leaders. They're just like us and they know it, but they tend not to advertise it. Our world is governed, and this is the key phrase that sticks with me, governed by a fellowship of amateurs. And so I come back to this idea of a fellowship of amateurs pretty frequently, especially when I'm trying to do things that haven't maybe been tried before in maybe the combinations that I'm trying them, is just to realize that everyone's making it up as they go along, and that setting out on your own and doing something new or something a little bit scary is the only way to do it, really.
Absolutely. Well, that is very well said. And I think a lot of scientists will identify with this. You know, everyone has their heroes in science, and it's almost humbling when you get to meet them and talk to them, and you find out in that moment that they're real people. You know, they make mistakes. They struggle with things. They've been wrong. And I think this fellowship of amateurs, I love that phrase as well, really sums it up well. And I think it has great applications in science. You know, no matter how accomplished you are, there are things you don't know and you will never know. You're never going to have all those answers. So wonderful piece of advice and inspiration, something to leave our listeners with today to think about. Now, can you tell them how they can reach out to learn more about you and your research?
Well, as you've probably guessed, the best place to reach me is on Twitter.
Wonderful. And
I am at K.E. James on Twitter. And you can also find my email contact information on my website, which is kejames.com, which you can also link to from my Twitter account, from my Twitter bio. And if anybody doesn't use Twitter and I guess doesn't want to visit my website, you can also email me at drkejames@gmail.com. James@gmail.com. So it's DRKE James@gmail.com.
That is perfect. Well, Karen, thank you so much for joining us today and sharing a piece of your story.
Well, thanks a lot. It's been fun to talk to you and to travel again through some of these stories myself.
Absolutely. It was wonderful to chat with you today. And thank you to our listeners out there. We appreciate you joining us. And we'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.